Thank you for tuning in to tonight's Sea Week event. It's our fourth webinar, which tonight puts the spotlight on the invisible impact of microplastics in the marine environment. We are putting the spotlight and the microscope on a pervasive pollution, the effects of which are still not known. I think tonight is going to be interesting, perhaps a little challenging, perhaps even a little depressing, but it will be extremely interesting, so stick with us. I'm Alison Balance, I'm your MC for tonight. I'm a natural history writer, I'm a broadcaster, I'm a diver, and like all of us here, I live in a world dominated by plastic. It's ubiquitous and immortal, and its use has consequences which we are only just beginning to discover. But before we jump into an ocean filled with plastic, let's start with a karakia. Tokua te wairua kia rere ki ngā taumata. Hai arihi i a tātou mahi. Me tā tātou whai i ngā tikanga a rātou mā. Kia mau, kia ita, kia kore i a noro, kia pupuri, kia whakamoa, kia tina, tina, hui e, taiki e. In translation, that means allow one spirit to exercise its potential, to guide us in our work, as well as in our pursuit of our ancestral traditions. Take hold and preserve it, ensure it is never lost, hold fast, secure it, draw together, affirm. Kia ora. Now, welcome back if you joined us for any of the previous Ngā Kōrero webinars. And if you're new to this, a very big hello. We'd love for you to introduce yourselves to us in the chat. I see that some of you are already doing that. We're telling us where you are. We love knowing where you're tuning in from. Everyone who's doing the webinar tonight, we're spread across the motu, so tell us where you are. The chat box is accessed via the text bubble icon at the bottom of your screen, which should bring up the chat panel over on the right-hand side. Hopefully that makes sense. The chat box is at the bottom. Click on that and it'll bring up the panel on the right-hand side. Now, a bit about Sea Week, Kaupapa Moana. It's New Zealand's annual national week celebrating the sea, Te Moana. Now, while Sea Week's pinnacle of physical events has already occurred at the beginning of March, and I hope you were out there being part of it, Sea Week's year-round mission is about exciting and inspiring all New Zealanders to renew their connections with the sea, which is why Sea Week is hosting this Ngā Kōrero webinar series, exploring ocean kaitiakitanga in Aotearoa, New Zealand. We want to foster better communication about various ocean-related topics. So we're highlighting the diverse connections and interactions we all have with the sea. We're reminding ourselves that our daily lives are intertwined with the health of our ocean environment. So take this as an opportunity for all of us to engage, to learn about our impact on the environment and the actions we can take to safeguard the ocean. We'd like to say a very big thank you to our longtime supporters of Sea Week, and in particular, those that have made this webinar series possible: the New Zealand National Commission for UNESCO and the Live Ocean Foundation. Thank you very much. Now, don't forget, the session is being recorded, so you can come back later and watch again or share it so others can watch later. And just so you know, we plan to finish by 8:30. So, microplastic. Microplastics are a complex issue. We're discovering they have impacts on human health and the health of the marine environment. Our experts are going to shine a light on the often invisible impacts of microplastics in the sea. They'll tell us about the incidence of microplastics in New Zealand marine species, the potential impacts of plastic debris for marine biosecurity, what happens to a fish that eats plastic, and what's the connection between our laundry and fish health. Let me introduce tonight's fabulous lineup. First, we'll hear from Dr. Olga Pantos. Olga is a research scientist at ESR, the Institute for Environmental Science and Research. She works in the Water and Biowaste Group and co-leads the AIM2 project. So that stands for Aotearoa Impacts and Mitigation of Microplastics. That's the two M's. She'll be setting the scene for us, as well as talking about her research. Next up will be Dr. Anastasia Zyko. Anastasia is an experienced molecular ecologist. She's co-leader of the Cawthron Institute's Marine Biosecurity Toolbox program. This is a project aimed at protecting New Zealand's marine environments from the impacts of non-native species. She's also a senior lecturer in the Institute of Marine Science at the University of Auckland. Anastasia will be telling us about biosecurity risks associated with marine plastic pollution in Aotearoa's coastal waters. Our final speaker will be Veronica Rotman. Veronica is a marine scientist and a science communicator. She's a member of the Sustainable Seas National Science Challenge Stakeholder Panel. She teaches marine science and aquaculture at a tertiary level to Mure Whenua in the far north. And her master's degree research looked at the impact of plastics on New Zealand fish. We'll hear from Olga, Anastasia, and Veronica, and then I'll ask them your questions. Please send us your questions at any time. Use the Q&A function, which is also on the bottom of your screen, very close to that chat function. You can ask your questions anytime, but we will ask them once all the presentations have been finished. So to start us off, we are very privileged to have Dr. Olga Pantos to give us an overview on microplastics. Take it away, Olga.
Okay, hopefully you can see my slide.
Ka pai?
Looks great. Excellent, good start. Kia ora koutou katoa. So I want to talk to you about microplastics, what they are, where do they come from, and why we're worried about them. I'll then share a few results from the AIM2 project. There's a huge amount of information around microplastics and plastics and the impacts that they can have. So I'll try and keep to the basics, and then Anna and Veronica will cover some of the other details, and hopefully we can fill some of the gaps through our discussion at the end of the presentations. So the first synthetic polymer, which we commonly refer to as plastic, was developed in the very early 1900s. But it wasn't until the 1950s that plastics went into mainstream use. Since then, the annual production and use has grown exponentially, with more than 368 million tonnes being produced in 2019 alone, and it's expected to grow to 60 million tonnes by 2025, which isn't that far away. Right now, more than half of all plastic ever produced occurred within the last 20 years. These numbers do not include synthetic fibres, and so the actual number is much greater than this. And this isn't a small amount that we're missing out from here if you consider that 60% of our clothing is now made from synthetic fabrics. So plastics are used in every aspect of our daily lives. The predominant use is in food packaging and then building and construction. It's used in our cars, our electronics, our clothing. You look around your house and you'll see it everywhere. Agriculture also uses a lot of plastic in the production of food. So it's not just the packaging at the end, it's all the way along the life of the production of the food. And then there are lots of other uses for the plastic, such as medicine. So at the end of the life of the plastic item, there are three main fates, and it depends upon the country or region you're in as to what fate this plastic will meet. So those three fates are landfill, incineration, and recycling, with the majority of plastics ending up in our landfills and only a very small amount, about 9% globally, actually being recycled. So how much plastic is going into our oceans? You'll have heard about the predictions of there being more plastics than fish in our oceans by 2050. Recently, these figures have been reassessed, and it's suggested that these original numbers were underestimated by about 50%. So now there's an estimate that's actually 19 to 23 million tonnes entered the oceans in 2016, and that's 11% of all the plastic produced that year. And these estimates are actually concentrated on the larger plastics, so it isn't clear what the levels of small, macro and microplastics are, and even less is known about the levels of nanoplastics. over time, the levels of the small stuff will increase relative to the large stuff as the large stuff starts to break down. So there's a time lag between those big plastics entering the environment and them turning into the microplastics and that microplastic pollution that we're seeing there. And everybody will have heard about the plastic soup in our oceans, but in reality, the fields that our crops are grown in have between 4 to 23 times more plastic pollution than our oceans. And it's an area of research that's only relatively recently been focused on. And of course, all those plastics that are sitting there in our soils can gradually make their way out to the sea over time. So one of those properties that make plastic so popular and used for so many things is its durability. It can't be digested by organisms and used as a food source. This means that plastic never degrades. Instead, it fragments into ever smaller pieces until it can't be seen by the naked eye anymore. So only 5% of the plastic in the ocean actually floats on the surface of the water. The rest drifts deeper into the water column or lies on the seabed. It can be consumed by creatures, both when it's in the water column and down on the sediments, and it will be continuously cycled through the ecosystem. So every time it passes through the system, because it won't be digested, no, it's another opportunity for it to cause harm. So before we go any further, it's really important that we first understand what plastic really is. So the Oxford English Dictionary definition of plastic is a synthetic material made from a wide range of organic polymers such as polyethylene, PVC, nylon, etc., that can be molded into shape while soft and then set in a rigid or slightly elastic form. But when we're talking about plastic pollution, we're talking about not just that polymer, but the chemical additives that are added to the base polymer to allow manufacture and to give the end product the desired properties. And so the reason we consider plastic as the polymer plus additive is that these additives also play a role in the impact plastics can have on the environment. So what are microplastics? Microplastics are defined as plastic particles less than 5 millimeters in their widest dimension. They're split into two subcategories. You have primary microplastics, which are those plastics that are deliberately made to that size to suit their purpose. So for example, microbeads like you get in personal care products, although they're banned in New Zealand now, and glitter. Then there are secondary microplastics, which are produced as larger plastics, produced from the breakdown of larger plastics. Plastics will continue to fragment to smaller and smaller pieces. And once they get down to less than 1 micron, they're referred to nanoplastics. So these size range definitions are a little bit variable between research groups, but that upper range of microplastics at 5 millimeters is a bit more consistent. So you might hear a slight variation, but they're all just small plastics. So what are the sources of plastics? Anything made from plastic. So we're looking at some of our different areas. We've got urban use, building industry uses a lot of insulation, pipe work, the cabling, it's all covered in plastic because of its property of being a really good insulator. Plastic litter makes its way out to the oceans through stormwater. Plastic has been great for transport because it makes everything lighter, so you're using less fuel. So our wastewater is a massive source of plastic. Of larger items. If you look around your kitchen, you use a lot of plastic. The inside of your dishwasher is like introduced during the packaging process. or if with seafood, I'm sure you'll have heard of the presence of plastic in mussels and other creatures. So we're eating the food, and what goes in must come out, and it heads off to the wastewater treatment plant. The wastewater treatment plant itself uses a lot of plastic in its processes, so there's another source of plastic. So as wastewater treatment plants aren't designed to remove plastic, you're seeing a lot go through, and some is trapped and removed from it. So these are just some examples of the master's project by Helena Ruffel a couple of years ago. And these are some of the things she found. So there are examples of glitter, large amounts of glitter, bits of dishwashing sponge, and fibers and fragments, all kinds of things, and all kinds of different types of plastic. And these images are from the water going into the wastewater treatment plant and coming out of the wastewater treatment plant, which is being released out into the open ocean. So you'll see from the graph that the polyester is one of the most common polymers found in wastewater from these sites, and that's synthetic clothing. So landfill, it's obviously a place where a lot of our plastic waste goes to, and it's still out in the environment, it's just out of sight. And the Fox Glacier landfill disaster was a real reminder of the fact that it's just hidden and it's not gone. So as all your waste goes into the landfill, it gets compressed and broken down, and it's lots of things mixing together, and you get fragmentation of the larger plastic items, creating nano and microplastics. And in managed landfills, they actually take this liquid that oozes out of the bottom, and they send some of it back through your wastewater treatment plants for treatment. So there's another source out to the environment via your wastewater treatment plants. A large amount of plastics are used these days in food production, from clips and twine to netting and plastic mulch to help with water retention and pest control, which is done to help reduce the need for pesticides and help reduce the demand on our water. Some of the less obvious sources of plastic include slow release fertilizers. I don't know if you can see within the roots of this lettuce, there's little pellets. So some of those are still using polyurethane, polyethylene for the coating of the pellets. So they're immediately microplastics. They're less than 5 millimeters, and as they have done their job, they break down into nanoplastics. And another source to the soils is the use of domestic green waste compost. There's a large amount of plastic going into our green bins with our food, either intentionally or unintentionally, especially these plastic food stickers, which seem to be very sneaky and get through. And of course, marine activities, from recreational boating to commercial fishing. The lightweight, durable and resilient nature of plastics make it ideal for use in the marine environment. But once we start looking, you'll start to see how much plastic really is all around us. You know, from our decorations around the house and at Christmas, to our crafting, our food production in the home, food preparation, our cosmetics, weed matting. These clips were by a river that had weed mattings for stabilization of the riverbank, but you could see them breaking down in situ into microplastics. For some reason, somebody felt that these lilies weren't pretty enough, and they put glitter in the center of them to make them prettier. Fishing lures had plastic glitter within them to attract the fish. Now, even down to our shoes, we know they wear out, that it goes somewhere, and it's the plastic out in the environment. So why are we worried about them? Although plastics are generally considered biologically inert because nothing can utilize them as an energy source, they're not inert. They can have a huge range of impacts on organisms and consequently on the ecosystem health as well. So there are a range of factors that can affect how a plastic can cause damage. So the effects are dependent upon their associated chemicals. These can either be inherent in the plastics, those additives that are put in to make the item do its job, or they can be acquired. So they're chemicals out in the environment that can be taken up by the plastic, because the properties of the surface of the plastic mean that they are quite good at absorbing those chemicals in high concentrations onto their surface. The base polymer itself can have an effect, and it can also influence the associated chemicals. Their size, as they get smaller, they can pass through membranes and have a potential impact at the cellular level. So the microbes that become associated with them, this may vary depending on the associated chemicals and the type of the polymer and the age of the plastic. If it's a brand new piece of plastic or one that's been floating out in the ocean since the 50s when plastic went into mass production, the older it is, there are changes to the surface properties, which in turn can affect the chemicals that become associated with them and the microbes that become associated with them. And of course, the organisms that they interact with affects what the impact may be. And there are probably many other factors that we've not yet identified. So I'm not going to go into any detail about potential impacts of micro nanoplastics, as there's just far too much to cover. So I'll just go through and list them quickly. But Anna and Veronica will talk more about some of these in their talks. So they include physical effects such as blockage, nutrition, and starvation. So those things that we see, turtles and large megafauna consuming plastic, and it causes problems. This all happens at the small scale as well. If you're a little worm and you're eating plastic, relatively, a microplastic is still big. It can cause inflammation and infection, physical damage, reduced motility, reduced biomass and growth efficiency, which can affect reproduction. And biological and biochemical effects, such as the accumulation of other contaminants, increased uptake of other contaminants, endocrine disruption, having reproductive impacts, disruption of cellular processes, failure of organs, and behavioural changes. There can be changes to those microbial communities, and those microbial communities play critical roles in ecosystem function, from nitrogen cycling to carbon fixation. And Anna will talk about the biosecurity risk that plastics can pose in the marine system. In wastewater treatment plants, they can act as mini rafts for human pathogens to pass through the system and out into the environment without being treated properly. And there's increasing evidence showing that plastics provide a sort of perfect storm. They provide a surface, they have chemical contaminants that can turn on antimicrobial resistance and promote horizontal gene transfer. So this can cause an increase in virulence and resistance in these microbial communities. And this is of particular concern when it comes to plastics coming through wastewater treatment plants, because we know there are lots of human pathogens there. There are lots of chemicals, lots of antibiotics, and lots of plastics. And so we need to understand this area much better. And there's a lot of work being carried out at the moment on that. So as part of the AIM2 project, we've been looking at levels of microplastics in marine, freshwater and terrestrial environments in Aotearoa. So I just wanted to share briefly some of the results to highlight that even though Aotearoa has a low population density and is sort of considered hidden away down here in pristine corner of the world, plastics are most definitely here in huge numbers, but then may not be visible to the naked eye. So we looked at microplastic at 11 beaches in Northland in summer and winter. The microplastics were split into two categories. We looked at large ones, which were more than 300 microns, and small ones, so less than 300 microns. There was a great amount of variation between the sites, with some where no plastics was found, which was great, and the levels were low. But when we looked at the small microplastics, at less than 300 microns, we were seeing up to 9,062 particles per kilogram of dry sediment. And it's those high production plastics, the polyethylene, polypropylene, and PET were predominantly found there. So we also looked at, oh, that's gone a bit funny. We also looked in the folk catchment in Auckland, in upper and lower reaches of it. And we saw much greater levels of the large microplastics and really astonishingly high levels of the small microplastics in these sediments. You can see from the graphs that we've got the same high production polymers present in the large microplastics, and a different selection of them found in the small microplastics. So the levels of large microplastics we saw in the FO are very similar to the levels found in mangrove sediments in Singapore and estuarine sites across the Waitemata Harbour. But they were 6 times greater than the levels that were seen in Auckland beaches in a study done by Jamie Bridson at Scion a few years ago. So for both locations, we're unable to compare the small microplastics because very few studies have actually looked at particles that small because of the challenges of doing the analysis. It's a very labour intensive process. So then some of the other things we've been looking at in plastics in biota. So there was a study that I was involved with Karen Stocking at Massey University, and we looked at the stomachs of 15 common dolphins which had stranded and died at 3 locations across Aotearoa. A wide range of polymers were found, both fibres and fragments. The usual suspects were found: polypropylene, polyethylene, ABS and PET. You'll notice that PET only made up a small percentage of the fragments that we found, but the majority of fibres. The PET is more commonly associated with drink bottles, but it's also the polymer that polyester fibre for fabrics is made from. So these fibres are most likely originating from wastewater effluent, and this corresponds to what Helena found in her wastewater study that I mentioned earlier. So we've also been looking at mussels across the country. This is just a brief example of what we're seeing. These mussels were collected from 3 sites within the Marlborough Sounds. It's an inner, middle and outer point along a sound, and we're seeing a very similar pattern across the country, with those high-use polymers being the most common. So we're also looking at different bivalve species that inhabit different ecological niches as well, because there may be some difference in the polymers they're associated with because of the differences in the polymer density, if they're floating in the water column or they're deposited out in the sediments. So as well as those, we're looking at the marine surface waters. We've done a series of trawls in different places, terrestrial soils and compost. So we hope to be able to share those results over the coming months. So notice I'm a little bit over time, but I just would really like to acknowledge and thank all the people that have helped us with the collection of those samples that I've just shown you there. The work was just made possible by their help. And that's me. Thank you very much.
Kia ora. Thank you, Olga. That was extremely interesting and quite confronting, I have to say. Lots of things to think about. In the meantime, next up, we have Dr. Anastasia Zyko. Anastasia, take it away.
Thank you. I will try to share my screen now. Just a moment. Does it work? Can you see it? Okay, so kia ora koutou, and today I'm going to talk about maybe not that obvious impact of plastic pollution, the biosecurity aspect of it, marine biosecurity. So why marine biosecurity? First of all, because it is a well-recognized global issue and a threat to marine ecosystems worldwide. And it refers to organisms that travel with some human aid from one region to another, and they manage to establish their populations, and they might pose some risks to the local biodiversity, ecosystems, economies, and even human health and well-being. And also as part of a biosecurity team at Cochrane Institute, we work on the different aspects of marine biosecurity, trying to understand better what are the ways how these organisms are getting into our ecosystems, what are the implications of these new invasions, and what are potential threats of that these species can cause to our environment, and also how we can mitigate these risks. So before I talk about the connection between plastics and marine biosecurity, I want to introduce the concept of marine biofouling to you if you don't know what it is yet. So if you immerse any substrate, like literally any material into water, into seawater, in a very short time, a range of organisms will occupy this substrate and will build up over time and form this biofouling, that is a dense community of different species. And it starts forming like from the very first hours of after immersion, and within a few days you will see this very thin slime layer formed by microbes, bacteria, small algae, and larvae of larger organisms. But in a few weeks or months' time, depending on the location and the season, you might get to this very dense growth with like a multiple species accumulating on the substrate, like in multiple layers. And it may contain even larger organisms such as mollusks, mussels, oysters, even crustaceans and algae and other species. And this is a natural process. But because we humans, we introduce a lot of artificial materials into our seawater. And we build new structures, marinas, we deploy buoys, and of course, vessels, ships that also have like a large surface altogether that is floating in the water every single second. And all these surfaces, they actually provide this potential surface for those little organisms to settle and accumulate. And this is the most important vector of how invasive species and unwanted organisms can get from one place to another and even cross oceans and get from one continent to very remote locations where they wouldn't be able to reach by themselves. And this is how most of the marine invasions happen. But now what we see, and you have heard from Olga's talk, that apart from all these multiple structures that are already there in the sea, we also have this another substrate that we created and that is accumulating over the last decades. It is plastic. And it is persistent, and it is there for a long time. And of course, it is also a very attractive substrate for all those little creatures to attach and thrive on it. And also, it is a new way for them to travel around. And there are evidences that some species could travel across the ocean. For example, following the Fukushima tsunami and a big amount of debris that were deposited into the sea, there were many organisms that are naturally present in Japan waters that traveled across the Pacific Ocean and were found alive in the western coast of the North America. And also the same happens everywhere. There are like species from Europe are found in Arctic islands, and they travel there on the plastic bottles. So in a few words, what we have now, that in addition to all those already existing surfaces, artificial substrates that we have in our coastal environment, in our marine environment, we have this additional plastic debris that are floating around and that also serve as vectors for invasive species to travel and raft and get from one place to another unnoticed and unmanaged. And although we know that it is happening, it is still pretty much not clear if there are any specific pattern how the species accumulate on plastic. How long does it take to grow, or like for different species to accumulate on different types of plastics? If there are any specific preference for particular polymers, if there are any specific additives in plastic that would promote growth of some invertebrates or some pathogens. And all these need to be understood better so we could evaluate what is the actual risk of biosecurity risk of this plastic pollution in the water. And as scientists, we are keen to understand it better and actually to help mitigate it in the future and find solutions how to do it, how to actually we can avoid these additional risks that we put into our ecosystem. And how we do it. So scientists like to run experiments and like to collect samples. And that's what we actually do as part of this AIM2 project as well. And I will present you a few results and few experimental studies and results that we have so far, and also some studies in progress that we hope to have some exciting results in the nearest future and understand better the biosecurity aspect of and risks associated with the plastic pollution. So the first experiment was a pretty small laboratory experiment run in these custom-made aquariums that were specifically designed to hold tokens made of different plastic polymers that are very common in our daily life, and also one token made of inert glass as a control. And then we added seawater into those aquariums. And also we added larvae of 4 marine species that have different biological characteristics and also different larval settlement patterns and different larval development patterns. And also what is important that these 4 species, they are known to be marine pests either in New Zealand or elsewhere. So they are kind of good representatives of those species that we expect to accumulate on these plastic pollutants. And interestingly enough, we found that most of those species, they in fact selected these 2 plastic polymers, and they prefer to settle on them. So there is a selectivity, except for the oyster that was pretty much indifferent, and they didn't like mind where to settle. Also, we saw that there was a specific pattern of those larvae. They were not settling all over the tokens. Some of them, they tried to settle like closer to the water surface. Some of them, they accumulated more like along the edges of the plastic pieces of plastic. Some of them, they accumulated in the center. So it's not like a random process apparently. And in fact, there is a selectivity in those communities that form on those artificial plastic substrates. Then we thought, okay, you know, with this quite intensive wave of new solutions to the substitutes for the usual plastic polymers, and all the novel types of custom-made polymers that are supposed to be more ecologically friendly and biodegradable. We thought, like, if we use some of those new and custom-made polymers and see if there's any difference in settlement of those different species on these new materials. So together with the Science Research Institute, we created this custom-made substrates made of biodegradable polymer that was also augmented with the oyster shell waste from the aquaculture. And it is all natural, it is like biodegradable, and we added different amounts of oyster shells into those plastic polymers from like low concentration to high concentration. And then we applied molecular analysis that allows us to look into very small like details of the community composition, even like detect species that are not visually detectable, and characterize bacterial communities and small like larvae that are not identifiable by microscope. So basically we are using the same technology as the COVID detection workforce is using, for detecting those small fragments and fingerprints of the virus from the environment. But we are trying to use it for the entire community characterization. And by using this method, we were able to we followed the growth of the biofouling over 18 weeks on those panels. And on these graphs, you can see these colored bars indicating the non-indigenous species. So actually, these are species that do not belong to New Zealand environment. And they were brought to our waters with vessels, with aquaculture, and they managed to establish here. So it is quite usual that we see a lot of them in the environment like marina, where we deployed our substrate. And interestingly, for the first 2 weeks, there was not much difference between the control and the polymers that were had added oyster shells into them. But after week 6, there was quite a difference in the amount of those non-indigenous species we found on the oyster-augmented polymers, and also the compositional percentage of different species was quite different from the control, and also the high concentration or oyster shell polymers. So it gives us a hope that actually those new custom-made substitutes for plastic, they may help to protect our waters from accumulation of those unwanted organisms if they are properly designed and tested. And for example, if they're used in the engineered structures in marinas or in aquaculture facilities, they may help to reduce the accumulation of those unwanted species in those environments. Oh, sorry. And then we had yet another experiment that was run as part of the AIM2 project, and it was a nationwide deployment where teams from different institutions, from different locations participated and put together their forces to deploy these custom-made structures. You can see it on the pictures. It is like a metal frame with these Christmas trees made of different plastic polymers, 5 plastic polymers used. And some of them, they were specifically treated to represent like a artificially aged plastic, that plastic that was in nature for a while. So we can do it by using some UV light, like applying some UV treatment on that. And these structures, they were deployed in 3 locations: in Auckland, Nelson, and Christchurch. And we followed the growth of biological communities on these structures over 1 year. So the bottom photos, actually, they show how the structures looked after a few months and like closer to 1 year after the deployment. So from these nice and white and clean plastics, you see this very heavy growth of different stuff on them. And again, we applied molecular methods to identify the communities, to identify bacteria, and try to detect bacteria that potentially can contribute to biodegradation of those different polymers. Also understand the chemical changes in those polymers with time, what happens to them if they are deployed in the sea and left there for a long time, and also mechanical changes if they actually degrade and change their properties. So the analysis of these samples is still in progress, and we hope to see some exciting results in the next few months. But you might also ask, so okay, so how does it relate to the actual life, to what we see in the ocean? So to answer this question, we then go, like using the information that we get from the experiment, we then go at sea and sample. And then we try to, first of all, understand what kind of plastics we can find in the marine waters, the microplastic accumulation, what types of microplastics, what are the hotspots of this plastic pollution around New Zealand, and also what are the invasive species that are found in the same locations and how they can be related to the plastic that is drifting along our coasts. And we were lucky enough last year to have 2 exciting expeditions in the Northern Ireland, Auckland to Northland, and also Fiordland, where two teams, Biosecurity and Microplastics, worked together, and we collected these samples from to learn more about the microplastic pollution around the coasts and also the invasive species that are found in the same locations. So we could relate this data together, put it together, and then predict better what are the risks associated with the drifting plastics and biosecurity implications of it. And also, what is another very exciting thing that these trips and research cruises gave us an opportunity to provide some outreach activities to the local schools and communities. So we stopped by the way, and we gave talks and some hands-on experiences to the local schools, to communities, to people interested, to talk about the plastic pollution, to show them what kind of plastic we found in the environment, like in their neighborhood. What is microplastic, how dangerous it is, how important it is to follow it and to think about it and care about it. And also we had an opportunity to showcase the molecular technologies that can be used to understand better the biology of those plastic pollutants and understand what are the biological organisms, the biological communities that are associated with plastics, and pests that can be detected on the marine plastic litter. And I would like to close my presentation with this saying by Sylvia Earle, that in fact, it is the worst of times, but it is the best of times because we still have a chance. So I think it is good. It is really a quite upsetting situation, what we observe now in terms of this plastic pollution in the ocean, but it is good that we understand that the problem there. And we are trying to find the solutions, and it is really important to keep this work going on and to educate people and to try to change their attitude and their understanding that we need to do something about it and find new solutions for substitute maybe plastics, to use it less where possible, and to substitute it with something more ecologically friendly in the future. And I also want to thank the amazing team of AIM2 project, and especially the deployment team, people from multiple research organizations around the country who helped to organize and set up this really very exciting and very complex experiment. Thank you.
Kia ora. Thanks, Anastasia. That was really interesting again. And you're right, very complex, trying to tease out some of what's going on. For all you watchers and listeners out there, thanks for the questions so far. Do jump into the Q&A tab at the bottom of the screen to add your questions, and we'll ask them at the end of the presentations. But for now, we have one more presentation, and we're going to delve a little deeper into the incidence and effect of microplastics on our kaimoana, our marine life, with Veronica Rotman. Take it away, Veronica.
Thanks, Alison. I'll just figure out how to do this screen sharing business. Cool.
Looking good.
Hopefully you can see that there. Kia ora. Ko Veronica tukuingoa no Aratana ho. Ko remarkables tukumonga, ko aro te awa, ko Aotearoa te iwi, ko Ngāti Hoa te whānau. Tēnā koutou, tēnā koutou, tēnā koutou katoa. My name is Veronica. I'm from the far south down in Aratan and working up in the far north with the Murewhenua. Shout out to any of the students. It's a privilege to be here on this panel with two incredible wahine who I've been fortunate to have referenced throughout my own research. So thanks for sharing your knowledge with us this evening. Now, to get right into it, what does beer tap water, human stool, salt, and now blood have in common? Well, they've all been found to have microplastics in some quantity. Just publicized last week, there was an average of 1.6 micrograms of plastic found in every milliliter of blood in some, I think, 17 healthy participants. And since the discovery of microplastics in these various mediums, interest has certainly increased as to how on earth they got there. So what's the problem if we chomp a microplastic here and there, little particle? As Anna highlighted, plastic acts as a vector to bacteria, pathogens, invasives. The scary thing is there is still very little research into the effect of plastic consumption on humans. Now, we do know that they act as a sponge to POPs, or persistent organic pollutants. These are industrial chemicals, pesticides, other synthetic pollutants that are present in the ocean from various means and cling to plastic. Now, there are also additives that Olga touched on. These are things like plasticizers, flame retardants, stabilizers, pigments, all sorts. And these are added to plastic to give their rigidity and various properties. Now, both of these are knowingly toxic to humans and can cause carcinogenic, immune, reproductive, neurobehavioral problems. Now, just to highlight quite how gnarly some of this tiny plastic is, there was a study that deliberately let pregnant mice inhale extremely tiny nanoparticles of plastic. And later, these particles were found in almost every organs of their little babies, which is not so nice. So ideally, we do not want these chemicals in our bodily temples. Now, microplastic size and sometimes shape mimics that of plankton and algae. These are the foundations that support the entire ocean food chain and the diet of the seafood-loving Homo sapien. So we've got bivalves here, like mussels, like oysters. These are filter feeders, and they literally munch to purify the ocean and ingest small particles along with their food. A 2018 study in the UK found 70 pieces of microplastic per 100 grams of mussels. There was a New Zealand study by researchers from Canterbury University, and they found up to 1.5 particles per mussel. And Olga mentioned earlier that her team found up to 4 fibers per mussel. Now, the problem with this is that we consume these shellfish whole. I very much enjoyed my bluff oysters last night, but of course we consume their entire digestive tract as well. So if there are microplastics within that, then that is a direct source of exposure. Now, ingestion has been reported in a wide range of taxa, from invertebrates at the base of your food web, zooplankton, all of those little bits and bobs, to elasmobranchs, or our big sharks at the top of the food web. It can occur directly through ingestion, or it can occur indirectly through consumption of contaminated prey. And one or more particles have been found in the digestive tracts of fish all over the world. Some fairly sobering stats there, with 95% of fish sampled in the Great Barrier Reef containing one or more particle. Now, the obstruction can provide the false feeling of a full tummy, but without the nutritional benefits. In fact, with some chemicals. Microplastics have been shown to influence the health of marine organisms through starvation, internal abrasions, blockages, toxicity, reproductive impact, neurobehavioral changes, just to name a few. The chemicals within or adsorbed to the plastic can also bioaccumulate up the food chain. Now, humans have this inherent ability to care about what directly affects them a fair bit of the time. So as emotive and heart-wrenching it is to display a picture of turtles with straws in their nose, if you can show that that straw is getting into your fish and chips, then policy change may happen a little quicker. So that led on to some of my own research. I wanted to know a bunch of things. So I went swimming in fish guts. We looked into whether or not hoki are ingesting microplastics in the wild, whether plastic is able to be retained in the snapper gut and how it affects it. Does ingestion affect growth and condition of the fish? And can it translocate into the flesh that we eat? Now, hoki is a deep sea fish, lives between 200 to 800 meters and is popularly known for its global use in Macca's fish products. Maybe a fish fillet burger. It is also one of our most commercially valuable fish. So I was quite intrigued. I sampled 20 individuals from 3 different locations. So from the West Coast, Cook Strait and Chatham Rise, dissecting their digestive tracts under a positive laminar flow hood, which helps to prevent contamination, which is a huge, huge problem with microplastic studies, and going through a series of chemical and mechanical steps until we had a dry sample of mostly inorganic material on a glass filter paper to be inspected under the microscope. So what did we find? We found 1 or more microplastics in the digestive tracts of 57 out of 60 hoki. The ingestion rate was higher than prior studies in Brazilian estuaries, the Spanish Atlantic and Turkey, but it was quite similar to that of the Great Barrier Reef. Various studies in China, Rapa Nui, and not too far off Fiji either. Similar average number of particles per fish. Now, Cook Strait had the highest ingestion, on average 6.9 particles per fish. And you could throw a guesstimate out there to say that could possibly be due to its proximity to anthropogenic activity, to pollution, to wastewater treatment plants. Plastics can enter as high density, and biofouling, weathering can alter the density and take it down to the deep where the hoki live. Now, these graphs here that took way too long to make are just fabulous, nerdy ways of saying that the majority of plastics found in the stomachs of and intestinal tracts of hoki were under 2 millimeters. They were black, white, and blue, and 90.1% of them were microfibers. Now, I'm going to come back to microfibers at the end, but I want you to keep that in your mind. So what about New Zealand's beloved snapper? How do microplastic affect them? So I carried out a 3-month experiment at Niwa up in Bream Bay, including small amounts of plastic in the feed of 150 juvenile snapper to look at various things. Now, there was a marine treatment that was put out into a polluted area of the Auckland Harbour for just over a month to accumulate a little bit of a biofilm as one of those treatments as well. Now, we looked at firstly growth of the snapper. Now, this was significantly compromised in the first half of the trial, and overall microplastic treatment fish had reduced average weight gain. For example, control fish had an average weight gain of 28.5%, and high treatment fish had an average weight gain of 19.4%. And other studies have found similar results on growth and also condition, which is a measurement of how chunky essentially a fish is. Now, we also looked at retention. So we looked to see how many microplastics could be retained in the gut after 4 days of no food. And on average, this was between 3 and 4 particles in high treatments. Now, why this is important is because time somewhat deems the severity of negative effect on fish. The longer the microplastic has to stick around the gut, the longer it has to leach these chemicals, these additives, these persistent organic pollutants. Also, the longer it has to potentially translocate into other organs or be passed up the food chain through predation. So the ultimate fate of microplastic particles really depends on their ability to be excreted or deposited in the liver, spleen, other organs via transport in the bloodstream. So a particle's size, shape, and surface chemistry really governs the effect on the fish. Which leads me to translocation into the muscular tissue of the fish fillet. So we found evidence of really small amounts of translocation in treatment fish, with an average of 1 particle per fish in the high treatment, and that's including 2 full fillets. So as much as we could essentially get off safely. Now, other studies have found varying results, and it is quite widely accepted that only the tiniest particles below 130 microns, or around 1/10 of a millimeter and below, are able to translocate from the gut into the bloodstream and to these organs or muscular tissue via endocytosis or persorption. So important to bear in mind as well that these were so tiny, I have been unable to put them through objective chemical analysis, FTIR, and we used brightly colored plastic, so they were the main method for determining this. Now, we also saw hyperemia of the liver. Now, we saw this in around 70% of high treatment samples, and this happens when blood floods to the liver and is a sign of induced stress, poor feeding, and low levels of fatty acids. So this was quite a surprise. Now, histopathology is a fancy way of saying the study of looking at really, really tiny tissues and cells under a microscope and figuring out what's wrong with them. So we carried this out looking at cross sections of our snapper intestines. So you can imagine that circle there is essentially just a cross section of an intestine. And I'll let the pictures do the talking to some degree, but if you see A here, it is a lovely, healthy control fish. It's also looks really, really healthy. Its intestinal epithelium is nice and well structured, healthy villi, nice and symmetrical. Now, as you'll see, as you go B, C, D, E, you get a bit of degradation. You get removal of the villi, you get all of these goblet cells forming congestion bits and pieces. And D and E were really seen in the more high treatment fish. Now, 60% of high treatment fish exhibited severe changes to the structure and architecture of the intestinal tract. And I'll just quickly mention that the intestinal epithelium is really critical location for absorbing nutrients and hence energy for growth. So if this trial had carried out for beyond the 10 weeks that it did, chances are you'd compromise permeability, efficiency, and the immunological response of the GI tract, which is not going to be great. So these images here are just zooming in and showing you some of the changes, and these words here are just complicated ways of saying not great. So that was kind of our snapper research. Now I'll just bring you back to microfibers really quick. Remember that 90% of the microplastics in my hoki's gut were fibers. And these have been found to be the major polymer by shape ingested by 46 seafood organisms globally in a recent large review by Kimura et al. So it turns out they're a bit of a problem. Now, they may actually be more harmful and toxic to animals. This is because they've got a really large surface area to volume ratio, which means they've got more area to absorb pollutants and hence potentially release them into the body of organisms. Now, zooplankton, remember that they are at the very base of the food web and they underpin basically everything. Now, they found exposing microfibers to zooplankton that they took longer to pass through than other shapes of plastic. They also found that the zooplankton exposed produced half the number of usual larvae or babies, and they were also smaller. Now, some awesome research by Scion found that 87% of microparticles collected around Auckland beaches and waterways were fibers. Third of these, though, important to note, were plant-based, so not all fibers are plastic. Now, where do these come from? They come from washing machines, wastewater treatment plants, as Olga highlighted, sewage sludge, ropes, fishing gear, atmospheric fallout. They're so light they can go up and come back down. They also come from tires. That's quite a significant contribution to the microfibers. Now, let's go back to washing machines. In 2016, University of Santa Barbara study found that 250,000 microfibers can be shed from a single fleece jacket after just one wash. We know that washing machines are a huge source of microplastic through wastewater treatment plant discharge. In New Zealand, they've got 82 wastewater treatment plants serving around 75% of the population, and that's discharged into the marine environment. Now, what can we do about this? We're all about some positive actions instead of feeling slightly depresso. So we can wash less. Perhaps you don't need to wash your jeans after every wear. You can wash cold. That's causes less degradation of fibers off your clothes. And if you wash full, then you will get less friction and hence less fibers coming off. Buy natural materials, so cotton, linen, merino, hemp. Don't use a dryer. That helps break up your clothing quicker. You can purchase various filtering apparatuses like Cora balls, which help to take the fibers out of your washing machine before they get churned down the drain. And you can also buy less in secondhand. So there's heaps of things you can do. Don't feel overwhelmed, just get on with it. Now, I'd love to finish by extending big aroha and a huge thank you to my niwa whānau, especially the Bream Bay crew and my supervisor, Darren Parsons. Ngā mihi nui ki a koe.
Kia ora, thank you, Veronica. Another very interesting project. Now we're opening up the floor for questions from the audience. It's not too late for you to ask your questions. Pop into that Q&A function at the bottom of the screen and fire away. We have some questions already from William, who wants to know, how does plastic pollution affect plants? And a similar question, so I'll go on to it, from Marin. What effect do micro and nanoplastics have on plants? And she goes on to ask, when microplastics fall to the marine floor, can they be taken up by plants? Are there actually any plants or animals that can safely break down plastic and neutralize its effects? So let's start with Olga, but perhaps all of the speakers might have an opinion on that. Plastic pollution in plants.
Yes, so this is an area of research that's only really been happening for the last few years because the focus has been on the marine system. They are seeing that plants can take up small micro and nanoplastics into them and transfer them through to the edible parts, so the fruits and the leaves, the bits we're eating. So there doesn't necessarily have to be an effect on the plant as it's taken up, but some studies have shown that it can affect the germination of the seeds. So it's being taken up just as the seeds start to germinate, and they're taken up by the little root system. They can get in there and prevent growth and proper root formation. So it's not exactly clear as to the mechanism that's causing this, whether it is the physical presence of the plastic within the plant, or those additives that are present within the plastics themselves. Was one of the other questions that are there any plants or animals that are able to deal with the plastics? I don't think I have heard of any plants that can. The wax moth caterpillar has been found to eat plastic bags, and it's thought that there's an enzyme within their gut that is able to break down the plastics. But yeah, it's been quite a while since I've looked at that study, and I can't quite remember whether they actually looked at the caterpillar poo, because one of the big limitations that we have at the moment is the technology to be able to look at the smaller end of the scale on the microplastic, nanoplastic side. So we're quite good at, we can manufacture nanoplastics, but actually, so we can manufacture them to use them to test things, but actually going into the environment, taking environmental samples, there are huge hurdles that we need to overcome there. So I don't think we know whether maybe they were just eating it and grinding it up and turning it into nanoplastics, which then couldn't be seen. But there may be enzymes that are in their guts, which are most likely to actually be produced by the microbiomes of their guts, that are capable of breaking down the plastic. And part of our AIM2 project is actually looking at microbes, fungi and bacteria that have the capability of breaking down plastic completely, so fully remineralizing it and turning it into water, biomass and carbon dioxide. With hopefully some good news story at some point, we'll be able to say we found something that will help us. And if there are any microbes out there capable of breaking down plastic, they're clearly doing it very slowly, or we wouldn't be in the mess we're in right now.
That's a lovely and depressing thought. Thank you. Would Anastasia or anyone else like to comment on that? Plants, any great solutions hovering out there that might eat up all this plastic mountain that we've created?
I think Olga has covered it mostly. So I know that there might be some also other marine organisms that could potentially like chew on different plastic polymers. But again, it's not like fully getting rid of the plastic and biodegrade it into, you know, harmless elements, but it's more like creating like microplastics, breaking down into smaller particles. Yeah, but the hope is there that if we can find and actually culture some of the microorganisms that can deal with these polymers, then there might be some good news in the future.
Anything you'd like to add, Veronica?
Oh, no, I'm good for that one.
Great. Okay, well, a question from, let me just peer off to the side on my computer screen. Peter wonders, how do the microplastic levels found in fish compare to terrestrial animals, such as sheep, beef, deer, and what about food like tofu? Does any of you know anything about that?
Haven't looked at beef, unfortunately.
Yeah, I think we might find that we've got great experts in marine microplastics, and they haven't paid quite so much attention to what's happening on land. I see no reason to think that they wouldn't be consuming plastic in the same way, would be my sort of rough feeling. Anyway, thanks for that question, Peter. Giono says, thank you for the great presentations. Presumably a requirement to manage this issue will be the ability to measure. Are there simple, rapid and scalable methods to monitor microplastic abundance and or characteristics in food or coastal environments? If not, are there promising lines of inquiry?
Yeah, it's definitely a challenge. We have limits of detection for size. It's very labour intensive at the moment, so you have to make that choice between, do I do enough replicates for it to be statistically usable, or do I spend a lot of time just concentrating on a limited number of samples? It's a massive area of research. the global community is working hard to find better methods, because we know that we need to be able to measure this more efficiently and quicker and more accurately. But as Veronica mentioned, with the fibres, a lot of the fibres that, if you're just looking and you don't go to the sort of high end of the technical side, if you're finding things that fibres, you can't necessarily assume that they're synthetic. There is a lot of natural fibres out there, even if you're looking at semi-synthetic materials, so things like viscose. When you're analysing it, because it's a semi-synthetic, so it's a natural product that's been a bit of chemistry in it, it's changed it a bit, so it's semi-synthetic. Not all the techniques, and probably the techniques that people have most access to, and it's a bit quicker, they can't distinguish between whether it is a completely natural cellulose or a slightly tweaked cellulose as well. But yeah, there's a lot of work being carried out to be able to do this, because I know quite a few students would really appreciate it if we weren't getting them to count all of this stuff, because it takes a long time. But we want to make sure we're doing it accurately and carefully as well. And being able to do that will give us better ability to monitor how policy and change, human behaviour is helping. We need to be able to accurately see that what we're doing is helping to bring these levels down.
I think there are a few developments in the field of microfluidics now, that they are trying to approach this problem, how to calculate and monitor microplastics from the aquatic samples more rapidly and more precisely. But I think there is no, correct me if I'm wrong, Olga, there is no like a standard protocol, standard method that can be used and applied widely. But there are some trials happening. Yeah,
and the risk from a plastic comes from those things that I listed. So it's the polymer type, the chemicals associated, the morphology. There's the things that I've forgotten now. But with some of the techniques to be able to prove that it is a piece of plastic, you can't answer all of those questions with the one technique, and some of the techniques are destructive. So we need something that brings everything together so we can find out all of those things, because all of those things really affect whether they'll cause harm to particular ecosystems or particular organisms. So we can't just do one simple thing, because we know it's there, but the risk from it is still not that clear unless we know all of those factors, all of those characteristics.
This is a kind of related question. Sorry, carry on.
No, I just wanted to add that that's probably why we are like doing a lot of our research like experiment based, because we are trying to mimic and like reproduce the situation, what we expect to see in nature, and then like control it slightly better.
A related question, which is to do with, the difficulty of sampling things as they get smaller, is from Danny. Is there a limit to how small microplastics can break down to, or do they just become vanishingly small? And at which point, what are they if they're not nanoplastics? And he asks, is there any research into the differing impacts of nanoplastics and smaller bits of plastic?
So that there are lots of people looking at the effects of the plastics at the different sizes. So the nanoplastics is a big focus, especially looking at the cellular disruption that they can have, because they can pass through membranes and into cells. But a piece of particle just 30 microns in size is small enough for the phagocytes within your lungs to ingest it or internalize it and take it into your system. So when you're getting really small, they don't quite follow the same behavior, and they'll pass through things.
Here's a question that's just come in for Veronica. What is endocytosis and persorption, and what was the higher treatment? I'm not quite sure about the last bit of that question, but could you explain endocytosis and persorption to us?
Yeah, sure. So these are the two mechanisms at which we think that microplastics may be able to pass through the intestinal epithelium. So endocytosis is basically just little cells called eosinophils that actually basically pick the tiny, tiny little microplastic up and transport it through the intestinal epithelium. But we think that that can only occur with particles under 10 microns, so really, really small. And then persorption is what's sought to allow larger particles up to kind of 130 microns or so. And naturally that would be like diatoms, ciliates permeating the little villi tip, the little tip that was being degraded, as we saw through our histopathology. They're essentially like nuzzling their way through persorption, and perhaps the shape and sharpness of the microplastic could influence that. And the question regarding the high treatment, so that was talking about the quantity of microplastics included in the feed of the snapper. This was 1.2%. Now, it was deemed environmentally relevant based on the backing of another paper that looked at the number of microplastics being consumed by juvenile salmon over in BC. But if I had more time, I would have gone and sampled prey of snapper, so green-tail mussels, cockles, etc., and tried to create a concentration based on that. So high treatment, 1.2% per of the feed.
Great, thank you for that. Polly writes, amazing and terrifying research. Thanks, everyone. So many food-related sources, both on land and in aquaculture. Do you know if we have any regulations in Aotearoa to counter that? Anybody got any thoughts on that?
There are no regulations as yet. I don't think any country does have any. There is a move towards it. I know California has introduced regular monitoring for micro and nanoplastics in their drinking water. So we'll see what the move is there. I think all of the water in California, drinking water, comes from surface sources, so reservoirs. But of course here, the majority of our water is coming from groundwater sources as well. And so far, there's very little work has been done looking at the translocation of microplastics through soils and down into aquifers. And their impact on the aquifer ecosystems, because although your water comes out nice and clear with nothing floating about in it, the aquifers that the water is coming from should have a very healthy and lively ecosystem with all kinds of little invertebrates doing a really good job of keeping the water clean. So we know that microplastics are having impacts on our invertebrates in our marine and freshwater systems, but we don't yet understand what impacts they could have on our underground water systems as well. So yeah, I think water seems to be has been brought into the spotlight to try and understand what's there. But of course, all our water is coming through plastic pipes. So how it is when it comes out of the ground might be quite different when it gets to you. But as for the food, there's lots of studies looking to see how much is there, how it's getting there, whether it's through the processing of it or the packaging of it, or if it's fresh food, how it's getting into there, but no regulations as yet.
Okay, there's a few comments to do with students and teaching these concepts with students. Jennifer said, how do you approach these concepts with students? Sophie asked, as an undergraduate student, are there any initiatives, organisations you would suggest to get involved with among New Zealand? And Pragna made a great comment that I had run a microplastics teaching session with students in year 5 and 6, sorting microplastics using jam jar and tweezers. She commented, Sustainable Coastlines has a great citizen science platform for sorting and categorising plastic. And Sarah behind the scenes says we can recommend the Sustainable Coastlines Litter Intelligence Program. So do any of our speakers have anything to add to that suggestion?
I think it's a great suggestion, yeah. I think sustainable coastline is probably the first point of reference where you should go if you want to participate in these initiatives. They're doing a great job.
Excellent. Well, thank you everyone for your questions. I have a final question for our speakers to try and perhaps end tonight on a slightly more uplifting note. What can we, as general members of the public, do to help reduce the occurrence of plastic in the marine environment? I'll start in the order that you've spoken. So Olga, starting with you.
Gosh, I never feel like I said anything terribly positive anymore. Become more aware of the plastic in your world. People just don't realise. I certainly didn't realise until, you know, a number of years ago when I started researching this, just how much is there, how much unnecessary plastic is in our lives. Because removing any of that reduces the chance of it getting to our oceans.
Great, Anastasia.
I would say maybe just to, you know, to continue Olga's thought that be more thoughtful about your consumption habits. And when you buy something, think about the where it ends up, and that it is most likely made of plastic, and do you really need it? And what you can do to preserve the nature and not getting it disposed. So just basically change your attitude and change your habits.
Veronica.
I think that I'm just going to stick with the microfibers. Yeah, just doing those steps, trying to buy less synthetic fibers. It's a good place to start. Yeah, and just being more aware, like these guys have also pointed out, you don't need to stop eating seafood. Mussels are still one of the most sustainable options, but we know that it's in everything, so taking it with a grain of salt, hopefully that salt's not full of microplastics too.
Thanks everyone. A huge thank you to tonight's speakers, Olga, Anastasia and Veronica. You were fantastic. I have learned so much. Daunting, but really inspirational as well. To you, our audience, thanks for being here and asking such great questions. And I'm very sorry if we ran out of time before we got to your question. Kia ora and thanks as always to Sarah, working away behind the scenes to make this all happen. You are amazing. If you can take a moment to please fill out the survey, which we've just posted in the chat room. We'll also be sending out a follow-up email, which will have this link, because we really want to hear what you thought about tonight's webinar so we can make the next ones even better. If you missed our previous webinars, they are all online. Find every one of them on the Seaweek YouTube channel or on the Facebook page in the video section. Tonight's webinar will be uploaded later there. There will be another webinar. Keep watching the Seaweek Facebook page to find out when and where. Let's have a karakia before we go. Onuhia, onuhia, onuhia ki te uru tapunui, kia wātia, kia mama, te nako, tia tinana, te wairoa I te ara takata, koiara e rongo, whakaeria aki kei ronga, kia tina, huie, taikie. Draw on, draw on, draw on the supreme sacredness to clear, to free the heart, the body, and the spirit of mankind. Rongo, suspended high above us, draw together. Many thanks for your company. Ata marie, good night, stay safe.
Kia ora.
Kia ora.